Welcome to Ganglion Analytics. Accessing Archive 379, Project DMD. Duchenne muscular dystrophy is a fatal disease characterized by progressive damage to skeletal muscles and early death, often by failure of respiratory function. This occurs from a mutation in the gene that codes for dystrophin, a protein critical for forming a complex that reinforces structural integrity in muscle fibers. Without dystrophin, muscle contraction damages the fibers beyond regeneration. Accessing entry 249, Axonopathy. Extracting entry. The DMD pathologies of muscle cells have been documented in high detail and rigorously studied, but this entry is going to focus on a symptom of DMD that has received less attention: damage to the nervous system. In a healthy human system, movement of the skeletal muscle is controlled by chemical activation at a neuromuscular junction, where the muscle is at a synapse with and activated by a motor neuron, which originates in the spinal cord and is controlled by the brain. Neurons communicate by sending electrical impulses through an axon, an extension that sends the signal to a synapse with the cell it communicates with. The axon is coated with sheets of myelin, an insulative substance that enhances communication speed through the axon. Recall that Duchenne muscular dystrophy results from a lack of muscular dystrophin, so the problem originates in muscle fibers, not neurons. How does damage in the muscle fibers cause damage in the nervous system? Specific DMD axonopathies in motor neurons have only recently been documented in studies such as the one logged below. The target neurons in this study are in two groups of nerves, the phrenic nerve and the hypoglossal nerve. These two nerves control muscles critical for respiratory function, failure of which is a common cause of early death for DMD patients. The pathology of these muscles has been well documented, but this study aims to identify pathology in the motor neurons. The subject of this study was the MDX mouse. This mouse has a specific mutation in exon 23 of the dystrophin gene. This mouse therefore cannot synthesize dystrophin and develops the phenotype for DMD. The target neurons in these mice were analyzed for abnormalities compared to wild-type mice. To identify damage in MDX mice neurons caused by DMD, the phrenic nerve and hypoglossal nerve were extracted from MDX and wild-type mice. The nerves were then processed, preserved, cut into slices, stained, and analyzed under various microscopes for axon size and myelin sheath thickness. To test if neuron count was different in MDX mice, which would signify degradation and neuron death, spinal cords were also extracted from MDX mice. The spinal cords were then cut up by section, sliced, preserved, and stained. These slices were analyzed under a bright field microscope. Imaging of the MDX nerves revealed that axon size was smaller than wild-type axons for both targets, and severely deformed myelin sheaths were observed in MDX nerves as well. Imaging also revealed abnormal densities and sizes of mitochondria within the axoplasm of target neurons in MDX mice. Lastly, when neurons were counted in target nerves, there was no significant difference in quantity between MDX and wild-type mice, suggesting that neurons were damaged, but they were not killed. What can we learn about axonopathy from this study? In the MDX model, we documented reduced axon size and damaged myelin sheaths. Myelination is critical for efficient communication speed, so the DMD phenotype also includes impaired nerve communication in the target nerves. As for axon size, we saw the MDX mouse nerves have a disproportionately great number of small neurons compared to large neurons. Large caliber neurons are critical for controlling large groups of muscle fibers. Healthy models utilize a combination of small and large neurons for optimal communication, so this observed trend is another contributor to impaired nerve-muscle communication. Abnormalities in mitochondria are also indicators of cell dysfunction. Size measurements indicated that mitochondria in MDX nerves were swollen, a common sign of mitochondrial dysfunction. Accumulation of mitochondria in the axoplasm, that is, the cytoplasm in the axon, is an indicator that healthy transport through the axon may be impaired. It can also be a compensatory mechanism transporting extra organelles toward the damaged neuromuscular junction. Lastly, the fact that cell death was not observed in the MDX model combined with DMD dystrophin deficiency occurring in the muscle fibers leads to the suggestion that the pathology spreads from the muscle and reaches the axons, but not the neuron cell bodies. So damage to neurons is serious but not lethal to these cells. These findings are examples of novel discoveries of Duchenne muscular dystrophy's effects on the nervous system. Now that the pathologies have been identified, it is imperative to progress research into DMD treatments that target the nervous system, particularly motor neurons innervating skeletal muscles. While treatments that target muscular damage and alleviate respiratory symptoms are well underway, the threat of respiratory failure will persist because of nerve damage. As progress toward a cure for DMD continues, this pathology in the nervous system should be addressed in the future. Extraction complete. Shutting down.